Good morning. Today we will discuss some of the important aspects of section cutting. Uh, Taking a good section is actually art and science, and let's discuss a few things which are related with section cutting. The first important thing, why we should go for a section. Now any section is going to reveal inside structure, composition, and that is going to help us to understand function and other parts which are related with anatomy of a particular part of a plant. This is going to useful for us identification of the plant, maturity of the plant, and so many other aspects also. So suppose you can say this is the stem, and we can have a section by two different types. This is one way where we can cut. This is longitudinal section, and this is the one which can cut at this particular plane. We can call it as a transverse section. Generally, transverse section is also known as a TS. And why transverse section is more important? The reason is that transverse section represents all part of that particular stem or leaf, and that's why it gives us clear picture about composition and overall information about that section. So that is the reason why transverse section is preferred over the longitudinal section or sagittal section. Now first we will discuss how to choose the sample. Okay, sample. Now in this case, if you really want to choose a leaf, it should not be too young or too mature. If you are choosing too young leaf, the midrib and other parts of that leaf may not be mature enough, so sclerenchyma or the lignified portion is going to be less, so you are not getting good staining. The color which you are going to observe in that section will not be proper. So see, this is a particular leaf, this is a typical leaf, this is particular stalk, or you can say petiole. This is a midrib, and these are the veins. These are the veins. This is actually lamina. This is lamina, and this is called as the apex or tip of the leaf. Now which is the ideal portion where you can take section is this part. Okay, so this part is there where the midrib transverse section which is a passing through midrib. So you are going to cut like this. So you will be taking the leaf and you are going to cut like this after a particular processing. So first step is selection of the sample. If you are taking section of you can say stem or root, make sure it is about 3 millimeter in diameter. 3 millimeter in diameter, so that the stem or root is properly developed, but it should not be more than 5 millimeter. Otherwise section cutting will not be proper, because if the section is too thick, too mature, it will give you some problem. Now you are going to take that section in the test tube. Add some chloral hydrate solution. So chloral hydrate solution is actually bleaching agent, and which is going to bleach certain colored pigments, and you boil that specimen in the chloral hydrate, and because of that you will have clear section so that surface preparation, either leaf or stem, will become soft, and section cutting will be more easy if you boil it for a particular period, and that boiling depends on condition of the plant. Most of the time some drugs are there like isabgol, which don't need boiling at all. Some drugs are there like fennel is there, coriander there. You need 15 to 20 minutes of boiling. But some barks are there, for example cinnamon bark, cassia bark, they may need prolonged boiling, calmic stem. So in that case minimum half an hour or more than half hour, 1 hour boiling also needed if the underground part is very thick and woody section is there. So there you need to boil it vigorously, but make sure you are not going to boil it too much, otherwise section is not going to be there in proper shape of that section is going to change or distort. So basically there are certain precautions. First thing, importance of transverse section. Second thing, how to select the sample, whether the leaf, stem, root, bark or any other preparation also there, you can select for the transverse section. After that you are going to boil it with chloral hydrate, maybe 10 or 15 minutes depending upon condition of that bark or condition of that leaf, and then you are going to observe under microscope. Now you can take a slide, put that leaf on the slide and make brand new, take a brand new blade should not be used, and with fine hand, like you are cutting in one stroke. You are not going to, supposed to cut like this, and within fine stroke you are supposed to take minimum 15 to 20 different sections, and like see this is the leaf and you are going to cut like this. Okay, so these 15 or 20 small sections you are going to transfer into the stem and wait for some time, let the staining reaction occur, and after that with help of brush you can take and mount under the microscope. Now this is actually beauty. You are going to look inside the plant. Now how particular leaf section looks like, maybe in a couple of coming videos we are going to study in detail plant by plant, but till now these are general points which you should remember while cutting of the section. And thank you. Okay.